Hi, my name is Ansley Foster, coming to you live from the Sullivan Greenhouse to talk to you about my dissertation work, Characterization of the Genetic Mechanisms Influencing Perenniality Variation in Arabidopsis lyrata. What is Arabidopsis lyrata, you might ask? Well, I have two examples right here. This is the Spiderstulin and this is the Meristem population. They're two populations of the same species, but as you can see, they grow very differently. Spiderstulin grows far more vegetative than Meristem over time. What we want to understand are what are the genes actually influencing the differences between these populations. Arabidopsis lyrata is a very good model organism for the lab because one, it's easily manipulated, and two, it has a completely sequenced genome. That means the genes that we manipulate in our experiments are going to be the ones that we know are going to be influencing the trait of interest, in this case, lateral shoot development. This is very useful for understanding how reproductive barriers arise between these different populations because Meristem and Spiderstulin grow in two very different climates. Yet, we don't understand the genes that are helping them locally adapt. Lyrata is also a model organism because it can help us understand how these plants allocate and acquisition nutrients and resources to grow either vegetatively or reproductively over time. We'll also be using Arabidopsis thaliana in my research to understand the differences in the life histories of annual versus perennial plants. A thaliana is the annual cousin to A lyrata, which is a perennial plant. A thaliana also has a completely sequenced genome, and it's also a model organism that can be easily manipulated in the lab, and that's why we're going to be using it in my dissertation work. The big picture question of my research is, what are the molecular mechanisms that are influencing adaptive variation in annual versus perennial plants? To understand this, we must understand that annual plants have one life cycle. What they do is push most of their nutrients and resources, such as nitrogen, phosphorus, undifferentiated cells, and tissues, to grow reproductively toward the end of their life cycle. They're going to then produce as much fruit and seeds as possible before the end of the life cycle. Perennial plants do not do this. They grow vegetatively and reproductively, and toward the end of their life cycle, they will push some of their nutrients to becoming reproductive, but they will also keep some of their shoots and leaves in a vegetative state because they're going to have at least one more life cycle. So to understand the differences between these two growth patterns, you have to understand that there's a trade-off between growth and reproduction. This trade-off allows the plant to then either acquire the nutrients, first acquire the nutrients and then allocate it as it sees fit for its particular amount of life cycles. And that could be 2 or it could be 20. The plant or more. The plant never senesces or dies at the whole plant level. So we're going to be using two different techniques to actually understand what are the key genes that are influencing lateral shoot development by influencing auxin transport. The genes of interest in my study are going to be PIN1, PIN3, PILS2, and BRC2. We're going to be using our first objective to look at QTL or quantitative trait loci that have been identified in previous research that we believe are influencing the differences in lateral shoot development. We will be taking CRISPR-Cas9 constructs and knocking down the protein function of these candidate genes. When we do this, we're then going to observe, because of their influence on auxin transport or lateral shoot development, whether that actually confers differences in the growth of the plant over time. We are also going to be taking perennial genes that are located on the chromosome 2 region of these perennial plants and putting them into knockout lines of thaliana, which have these same genes but on the chromosome 1 region. So we're going to be taking knocked out genes from the same plant, and we're going to be putting the perennial genes into thaliana lines, and then we're going to see if it essentially changes the athaliana plant over to a perennial plant. The implication of this research is vast. We know that climate change is a really, really real thing. And we know that plants, unlike us, cannot migrate as fast as we can. Therefore, they must be able to locally adapt to changing climates. So if we understand the genes that are actually influencing things like lateral shoot production, fecundity, and survivability, we can protect natural populations of plants and possibly in the future develop plants that are more adapted to altered climates. Thank you for listening to my research. I hope you have enjoyed.